Hello, my name is Leonid Peshkin. I'm the senior author on this paper I'm trying to present to you today. It recently came out. The title is On Standardization of Controls in Lifespan Studies. And the reason we felt compelled to release this study is what we call a life cycle of elevator speech science in the field of aging. It is sort of a satirical representation, but we seem to be going in cycles where a new theory of aging comes out, related intervention is tried in model organisms, and results are published, which contain certain measurements of health and health span, physiology, and things like this, but also lifespan. And lifespan is precious data because those are hard numbers. So then the theory goes out of fashion, sometimes becomes dormant or goes away altogether, and literature goes to oblivion. Of course, the way we feel the things should evolve is that regardless of corresponding theory and intervention, the data should be still reusable. So collection of all of the papers where different interventions are tried in a particular organism has high value to be analyzed together, perhaps analyzed by machine learning methods or modeling methods. And there are certain things which prevent it today. So that's the gist of our paper, and I will illustrate it using some example. Um, this study itself is not important, it's just an illustration. This is study where overexpression of SERT6 led to extension of lifespan in the certain strain of mice. So if you just contrast the intervention curve in red with control curve in green, it looks very impressive. It looks like intervention has worked. Of course, you could focus on the shape of a particular curve and ask, does this shape correspond to what we expect this distribution to look like, coming from our understanding of what are statistical dependencies in this data in general, and it might or might not look like natural lifespan data. What we are proposing to do then is to pay attention to exactly which mice, which strain, which conditions were used, and go and look at the literature where same animals were used. Now, what we're going to do is to add to this plot lifespan curves for controls, and controls only from all other papers in gray. All of the corresponding papers are cited here. And what becomes immediately clear is that apparently this effect is not so impressive. The so-called life extension in red looks pretty ordinary compared to other study controls. But control in this particular study was apparently pretty short-lived. Unfortunately, no matter where we turned, the life extension effect in the presence of this literature-wide control looked unimpressive. So these are just other examples. Here's curcumin, again in the presence of controls, and here's barbarian. Barbarian, um, both control and intervention lived much, much shorter than, again, same mice of same strain in other papers. So in this case, taking barbarian would be like taking some sort of drug which helped certain group uh, in a particular condition, but not healthy controls. For example, if particular therapy extended life of cancer patients, which would be, of course, great, it would not be an argument for all of healthy controls to follow this intervention to extend their natural lifespan. Rapamycin, which is the most cherished intervention, sort of stands the test of comparison to literature-wide controls, but again becomes least impressive. So you could ask, how does one analyze a lifespan curve with respect to two different criteria? One criteria is, does this particular curve look the way it is supposed to look? The other one, does this difference between control and intervention look statistically significant in the presence at the background of all of those other controls? And we found a way to calculate both of those. We additionally propose a data format, which makes it much easier to collect the data across all of those studies and contrast it. We created a website where you can go and submit your data, browse existing data, compare the data across multiple studies, and calculate those coefficients, which we hope is going to be very useful to this community to develop some good standards and reuse all of the data despite certain interventions going in and out of fashion. And I thank you very much for your attention. If you would like to know more about work in the field of aging by my group, you are welcome to follow this QR code. Thank you very much.